This is a production of Cornell University.
Yeah, thank you very much for Kelly's invitation and Joss's introduction. So because I had some misunderstanding, I thought like 4 of us will share the total 8 minutes. So I tried to make my slides as compact as possible. So only 3 slides. So this is the format we use for our soil and crop science lightning talks, only 3 minutes, 3 slides. So I would like to bring you to a totally different scale from the previous 2 speakers and also last speaker by Mike. My lab, our scale was actually very large. Well, I usually start my talk from this 2-dimension scale, in space and in time. So in space, we start from leaf scale all the way to the globe. And at the time scale, our research ranges from hourly all the way to the decadal scale. So keep this in mind. The research program, we try to answer 2 fundamental questions. Number 1, we try to understand how the natural ecosystems and also the agricultural systems respond and feedback to the climate change and climate variability, and how they respond to anthropogenic activities. Again, the scale is a major theme in my lab. So here, the second question I want to answer is like, how can we harness the best sensing technology to monitor the interactions of these agriculture system, natural systems with the climate change and the anthropogenic activities, and how we can utilize the state-of-art models, process-based models, to predict such interaction feedbacks in order to inform policy making. So here in the blue fonts, I tried to highlight some of those projects we conducted under these 2 major themes. So the research are funded by DOE, USAID, NSF, USDA, and CEDA, and also Cornell Atkinson, and also the CIPS grant challenges. The major 2 approaches, number 1, we try to use the remote sensing observations, mostly from satellites, but we also utilize onboard remote sensing and also the tower-based sensing technologies. So here I try to give you this example showing this layest fleet of NASA satellites, all the way from 1970s to beyond the 2030s. So we utilize different types of observations, starting from including optical remote sensing, thermal remote sensing, microwave remote sensing, LiDAR remote sensing, and also gravity remote sensing to really reveal different aspects of the Earth system, like for example, the soil moisture, vegetation health, and also like water availability. So I'm going to give you a few examples later. The second approach we use is called process-based model. This one example is the Earth system model or climate model. So such models have been utilized for the IPCC climate report every a few years. So why we use such models? Because it can help us understand the mechanism. So basically, we try to put the mechanistic understanding into mathematical equations and put into these type of models. So such models have like different components, including land components, ocean component, atmospheric components, and also like ice sheet, everything put together so that we can utilize such models to simulate the past and also simulate the future. So here, this is one typical figures which is the output of such models. Basically, these models can simulate the past. So during the past, where it overlap with the remote sensing technology, these data set can be utilized to benchmark such model simulations. So once we gain confidence of such model simulations, we can utilize these models to project the future to see, okay, under different economic or CO2 emission scenarios, what's going to be look like for the land system and also for the atmospheric system and also ocean system, everything. But here, my lab focuses on the terrestrial ecosystems and also the agriculture systems. So these are 2 major geospatial tools we use in my lab. Of course, on top of that, we also put AI to enhance the capability of these tools. Then here I'm going to give you some specific examples of the research projects. I really like to divide my research projects into this matrix. So this one dimension is field scale versus regional scales and beyond. Another dimension is what tools we use, including both observations and also process-based models. So at the field scale, we try to develop sensing technology and different observational data to monitor crop growth and dynamics governed by the genetic environment and management. So here, these two figures, the left one shows this two coupled tower systems that put the eddy currents measuring carbon, water, and energy fluxes, and also another system that monitors the hyperspectral and also chlorophyll fluorescence measurements, so that we harness the two measurements to understand the vegetation health and their response to different management, including irrigation, nitrogen management, and also different stress. In the meantime, we also developed the drone technologies that can put concurrent measurement of hyperspectral and also fluorescence measurements that can really look at many different targets, including different management treatments and also breeding plots, to look at their different functions. At the regional scale, we utilize really many different types of satellite data to monitor and assess the food, water, climate sustainability in both Africa and China. So the first example shows, the left figure shows, okay, the 2 decades of depletion, huge water depletion in northern western China. But this is a mystery of what was the causes for such a long-term depletion. So we look at these gravity data set, which give you information of total water storage. And in the meantime, we look at the land use, land cover change data set, and also the soil moisture and also evapotranspiration data set. In the end, we found it was really the crop expansion because of cotton growth that has dramatically expanded in the past couple of decades that led to this huge depletion of water, the total water storage. Because this is a very dry area and the cotton system really depends on the irrigation. But because it's very dry, there's no water. So the water should really come from the groundwater. So where the groundwater come from? That's from the glacier melting from Tian Shan Mountain. So very, very complex system. So here we try to use the satellite data set to really reveal what's exactly going on. And another example is like we try to utilize, again, different satellite data set to understand the impact of land restoration program in Ethiopia. So there has been a multi-million dollar investment from Ethiopia to put different the land restoration programs. But so far, we don't know what is the large scale, national scale impact. So we utilize different satellite data set to evaluate, okay, what is the productivity under drought? Turns out that such restoration program can actually improve the drought restoration for productivity. So such analysis can be done by the conventional approaches with like very sophisticated surveys that are labor intensive and very time consuming. So also at the field scale, we utilize models. So this is the collaboration with Mike Gore and Kelly Robbins. So we try to harness multiple remote sensing technologies and crop growth models and also AI to inform crop breeding. So one of my students were trying to run very sophisticated 3D radiative transfer models that benchmarked by the LiDAR measurement to see what is the best, the optimal corn structure that will have the best productivity for the future. And finally, we utilize both models and remote sensing data to look at the future crop yield, productivity, and also how the agricultural revolution can impact global carbon cycle. So one project we are in collaboration with the NCAR, which is National Center for Atmospheric Research, trying to understand, okay, what is the past driver, the green revolution, including the fertilizer and the genetics, can impact the CO2 amplitude increase over the past 5 decades, which has been a mystery. So these are a few projects I want to highlight. But in the meantime, we also work a lot on natural vegetation, including Amazon and Congo rainforest, and also the carbon and water coupling in across the U.S. So yeah, that's all I have. Thank you.
Any questions for you?
Yeah, would you let us know that what type of phenotypic data you can collect at the field scale?
Okay, so maybe Kelly can also chime in. Okay, yeah, so here we try to, the data set we collected, including LiDAR data, that's led by my Gort's group. And there are also the multispectral measurement from UAV. So LiDAR data was from like rovers, and also multispectral that's from UAV. And we also have some fluorescence data sets that is measured by UAV. So Kelly and I were just got the 6 grand challenge proposal that we want to collect more data in the G2F field in Musgrave. So of course, this is like an AI-based project. So the phenotypical data also, like, you know, we are looking at a lot of like the traits in the latent space, which probably we don't know what they are, but we other like more explicit traits includes say plant height, LAI, you know, leaf angles, or those of course yield. So all of these different information, but we're still looking at the data sets to, yeah. So that's why, you know, we're interested in like how you are trying to optimize the campus structure to get the maximize the productivity of apples.
So I have a question. So in advancing this, what are the bottlenecks, right? Is it quality of the imaging, the satellites? Is it the compute power? Is it the number of people developing new models? Is it the ground truthing the data sets? Or is it?
That's a great question and very hard to answer. Actually, to me, bottleneck is everywhere. So from the sensing technology part, I think the data uncertainty is really, really huge, especially for satellite and remote sensing. Because of course we do have a lot of data sets, but we don't know the level of uncertainties and how to trace those uncertainties. So that's something we have been working very hard on to trace those uncertainties, so that we can really link the satellite or sensing signal with the actual variable or trace of our interest. So that's very hard. I think for the ground remote sensing, I think we're bottlenecked by like the number of like the ground labels we have. I think that's quite hard to me. And another challenge is probably like analytical tools, like what is the best way to really harness remote the process-based models and also AI. You know, a lot of people are talking about like we should create like knowledge-guided machine learning, but you know there are a lot of different ways of how to do that in the best way and most efficient way. Of course, once we use like the deep learning or AI, the computational power is always a challenge. So I would say the challenge is everywhere. So a lot of work to do.
Yeah. Fantastic. Okay, many thanks, Ying.
This has been a production of Cornell University, on the web at cornell.edu.
Right.
Okay.
There's one slide that's not patterned correctly. Sorry, Joss.
Mike Scanlon, time everything.
Right. So I'm going to tell you guys today, our lab works on patterning, especially of leaf grasses. We want to know how leaves are initiated from the meristem and how they form these beautiful structures such as the blade, the ligule, and the sheath. We've done a lot of comparative analyses between eudicot and grass leaves. There's lots of differences in morphology. I'm going to focus today on these two, the differences in the leaf base. Whereas in eudicots, the leaf bases are usually non-sheathing, and they're accompanied oftentimes by these stipules, these lateral derivations, whereas grasses form sheathing leaf bases, as you can see better on this slide of real plants. A eudicot, this one has no stipule, but you can see the leaf inserts and it doesn't spread it all around the stem. Whereas in this grass plant, this is maize, we can see at the node, the sheath is overlapping. It forms a sheathing leaf base where there's a wrapper inside of a tucker. And we know that this happens in the sheath anyway, at the point of emergence. So if you're looking here at the node in this CT image, this computed tomography image, this is below where the leaf will initiate. Move up a little bit, you can see they're already overlapping as soon as they emerge. We know this from CT analysis, also from sectors. If you have a sector here, it'll mark both sides of the sheath, but not the blade. They come off the meristem, they come off the stem already overlapping. So how does this happen? So we've used a genetic approach to understand how these leaves wrap. And a long time ago, we worked on these two mutations called narrow-sheath 1 and narrow-sheath 2. Double mutations make a leaf that is very, very skinny. So this is a model of how leaves are maybe patterned into at least 3 mediolateral domains. The central domain, which gives rise to the midrib, shown here. The lateral domain, which is a large part of the leaf, and the marginal domain. We think our narrow sheath mutants delete this blue region here, this marginal domain, which is basically goes about halfway up the leaf blade and all the way down into the sheath. And as a consequence, these leaves no longer wrap. They look like pseudo eudicot morphology. This is almost like a petiole. If you do a cross section through this cartoon of the meristem, show these real leaves here. This is the 4th leaf from the meristem. Here's the meristem. Wild type, they overlap. The marginal domain is there. Narrow sheath removes this blue domain, and these leaves no longer wrap. They look more like a eudicot. And just quickly, you look at the expression. This is the shoot meristem with 2 leaf primordia. About a 2-week-old seedling. If you do a cross section about this level, you can see the meristem in the middle. This outer leaf, the 2nd leaf of the meristem, you can see expression shown with this dark blue area of narrow sheath at the margins, and it promotes growth from those margins. And this is actually the P1. The margins are not yet out from the meristem, but they're going to come out, and this marks where they're going to form. Okay. So we think the narrow sheath provides this outgrowth from the leaf margins. Now, in maize, there's actually 2 other WOX genes. This is genetics or maize genetics. There's 4 of these. There's only 1 in Arabidopsis. So we knocked out these other 2 called WOX3b, WOX3a, unfortunate names. And we found that triple mutations with narrow sheath 1, WOX3a make very, very, very skinny leaves. Look at the sides of the scale bars here compared to the narrow sheath, double mutant, and then the wild type. These are what the plants look like. They're very, very short. And they're very, very curly. And we think these mutations, narrow sheath 1 and 2, together with WOX3a, remove not just the marginal domain shown here, but also this large red lateral domain. All you're left with is the central domain and these triple mutants. And the growth happens from the upper part of the leaf. Okay. So the question is now, how do these margins wrap? What is the pattern of how they wrap? And it's been noticed that basically, if you look, again, here's the base of the sheath, there's the node, there's a wrapper and a tucker. If you do a cross section of a seedling, here's the stem, you see a couple leaves here. Notice if you draw a line through the middle of the stem, all the leaf margins that are derived from the right side of this stem will be wrappers, and all the ones from the left side will be tuckers. And this pattern is robust, it's decidedly non-random. And grasses. We can see the same thing happens in rice. And if you look at these as color-coded, Richie likes to color-code these. This is wild type, maize and rice. A non-random pattern of wrapping and tucking. All the margins coming from this side will be wrappers, from this side will be tuckers. So the question is, are there mutations that disrupt this pattern? And of course there are, I wouldn't tell you about them. So this one, you can see, everything was good until this one made a mistake. Wrong side, it's a random mistake. This next one didn't really go through the Mac to PC conversion very well, but you can see this one actually made 2 mistakes. And we find this in plants that are mutated in higher order WOX3 mutations. So if these plants are homozygous for WOX3a, and then homozygous for at least one of the other narrow sheath genes, heterozygous for the other, they'll give this random patterning of wrapper tuck. And the mistakes that these make are random. They get more and more severe as you go up the plant, make more and more leaves. So our mutants are in charge of this, which was a surprise. We didn't expect this, actually. And if you look at it closely, looking at some markers, again, this is the cross-section through the stem, wrapping and tucking, looking at this 4th leaf shown here, the expression of this boundary gene, CUC2, which marks very stiff cells that are very, very slow to divide. It's heavy in the tucker, not in the wrapper. You think this prevents this margin here from growing. In contrast, WOX3a, a gene which makes leaves grow from the margins, it's expressed very heavily in the wrapper part of the P4, but not the tucker. We think this promotes growth to make this thing wrap around. Okay, so that's what we're looking at now. So what Richie is doing now, a graduate student in the lab, is microdissecting with our laser microdissection scope the marginal domains of the wrapper and the tucker, put them in separate tubes, figure out what genes are being expressed here, doing the same thing for the lateral domains and the central domain, put them all in different tubes, make libraries out of them, and see what's going on in terms of gene expression. Okay. Yeah, that's what we're going to do next. So I'm going to skip this, and I think we have time for this weirdness. So there's another mutation we're working on in the lab. It's actually found here at Cornell by Emerson, person this building was made for, 1912. It's called ligulus. This, as the name suggests, this mutation removes the ligule, which is shown here, the boundary between the blade and the sheath. It's gone. So in wild type, we have these extended sheath margins. Look at these. These are epidermally derived, they're non-chlorophyllic, and they're very, very expanded in the sheath as opposed to the margin. We think this gives rise to the ligule. Narrow sheath removes the marginal domain, so these extended sheath margins are gone. The ligule is still there, but they're gone. We found that in ligulus mutants, and this was never described before, we see ectopic expression of this sheath margin extension way up into the blade, and it always happens on the Tucker side. So it's an asymmetric signal happening. So ligulus 1, unexpectedly, is also involved in patterning this wrapper Tucker. Okay, now it gets really weird. If you look at triple mutations, narrow sheath, which should have no margins, combine them with ligulus, somehow the margins of this triple mutant are restored. They're also ectopically displaced on the Tucker side only. So somehow, if you add these 3 mutations together, somehow narrow sheath can now grow margins again. So we suspect WOX3A is being patterned there to do that. And even more weirdly, if you look at these triple mutant leaves, the base of the sheath are oftentimes fused. They don't separate. No wrapper, no Tucker, they're fused. If you look at the microscopy, you can see these margins here, they can't decide whether to wrap or tuck, so they're fusing. They're all confused. And the last slide shows expression of WOX3A, which should be heavy on the wrapper. So these 2 leaf margins are confused. They don't know whether to wrap or tuck. And this one is fused. It's like a 2-headed monster. It doesn't really know which way to wrap or which way to tuck. So there's a lot of work to do to figure this out in the next couple of years. And I'll take any questions if there's time. Yeah, so we haven't, we get asked that question all the time. We have no done any experiments to see whether or not this wrapper Tucker mutations are affecting yield. We're probably never going to do those experiments, to tell you the truth. We won't do it. Yeah.
Do you have any ideas of like signaling that it's distinguished left, right?
So we know that the first leaf decides the pattern, whether it's going to wrap this way or wrap that way, and it's random. Like in Arabidopsis, the spiral phyllotaxy is going to happen, but it may be left or it may be right. So that first decision is random. We're trying to figure out now how WOX3 somehow in combination with ligulus can decide whether or not it's going to maintain that wrapping tucking relationship. We suspect that the signal to wrap and tuck, which side is going to wrap and tuck, is sent really early in leaf primordial formation. I didn't show everything that Richie's doing. He's actually going to be microdissecting the 4th leaf from the meristem all the way up to the 2nd leaf from the meristem. We think the signal happens really early and it sends down the plant. But that's what the big question is. What is the signal to make this wrapping tucking pattern? Yeah, that's a good question.
Sorry, this is kind of related to the question, but ever since QTL started to be detected, people have had this idea of breeding by design, where we would understand what all the genes the plant did, and then we would put the right ones in the right places, and then we would get higher yield. And to me, as a quantitative, it seems hopeless. But I think your answer to her question sort of was similar. But I wonder what you think about that. And like you study these things, but I don't know if this has an impact on yield or not. But does your research illuminate anything about the prospects for breeding by design?
So we're really interested in basic fundamental mechanisms of development, and I really don't even think about making more corn. I don't say that in my grant proposals, but I really don't think we need more corn. Sorry.
I think we need to lock the doors. Mike can't leave. We need to collaborate with somebody in this room.
Right. Oh, I've collaborated with Mike Gore a lot. So, yeah.
Any more questions? Yeah.
Yeah, can you ask what approach you use to identify those three, the narrative one, narrative two, and LD1? What approach do you use?
What
What approach?
do we use? So I deliberately, most of my talks are labeled genetics and genomic analyses, but today I just use genetics. We're using lots of single cell RNA-seq, spatial RNA-seq, multiplex in situs to try to figure out how these patterns form. And we're doing lots of reverse genetics. So yeah, we're doing trying everything we can actually to understand the mechanisms. We're really focused on the basic biological mechanisms.
So those genes are identified through reverse genetics?
Narrative1, Narrative2 were identified at Pioneer in a breeding stock. They won't tell us what the stock was, but they just arose. And then WOX3A and B, we made mutations by reverse genetics. It seems as if WOX3B, I didn't mention this, doesn't seem to have an effect in vegetative stages and might be affecting later. So that was done by reverse genetics. Narrative1 and 2 were identified by forward genetics. Cloned the conventional way, the old-fashioned way. Thanks.
One more question.
Have you seen or looked into how much of an overlap that Rapper and Tucker have? And does that affect how corn unfurls? I asked just because I've had difficulty with the thing in some genotypes before. Is that related at all to?
That's a great question. I mean, it very well could be. The degree of overlap varies. As you go from the base of the plant, from a grass plant to the tip, the size of the internode gets much, much smaller as you go up. So they tend to overlap more in the upper nodes and lower nodes, but they all come off the meristem, off the stem, in the sheath anyway, overlapping. The blade components do not. They wrap later. Great.
Okay, thanks, Mike, and thanks to all the speakers today.
This has been a production of Cornell University, on the web at cornell.edu.